Good afternoon and welcome to this afternoon session with Herantis Pharma, a Finnish-based biotech company developing treatments for diseases with a high unmet medical needs, so specifically Parkinson's disease and lymphedema. We're very pleased to have with us Craig Cook, who is the CEO of Herantis Pharma this afternoon. I'll hand you over to Craig now for a brief presentation before we'll close with a bit of a Q&A at the end. Craig, over to you.
Thank you, Adam. Delighted to be here. And thanks to everyone for joining. It's about 20 slides, so I'll go through fairly briskly so that we have time for Q&A at the end. Next slide, Adam. Usual disclaimer, next slide. Okay, so by way of brief introduction and background, I'm not clear how familiar everyone is with Herantis, so I'll just set the scene. So as Adam mentioned, we are a Finnish company based in Helsinki. We are listed on both the First North in Finland as well as in Sweden. And our assets originate from world-class research departments at the University of Helsinki. And moreover, our science and our research has been published in top-tier journals, including Nature Science, Nature Immunology, and JPRAS. So those are really top-tier journals, just testament to how good our research is. From a scientific perspective, our focus is very much on disease modification. So we're looking to slow, stop, or even reverse disease pathology. We have CDNF, which is cerebral dopamine neurotrophic factor for neurodegenerative diseases. And we have lymphactin, which is vascular endothelial growth factor type C, which is a gene therapy for lymphatic diseases. As I mentioned, our focus is on modifying the disease pathology. From a disease perspective, our focus with CDNF is Parkinson's disease, and our focus with lymphactin is lymphedema. Now, for both of these therapeutic areas, there has not been a lot of successful innovation. For decades, for Parkinson's, for example, the mainstay of therapy today is the same as it was 50 years ago, levodopa. And similarly, for lymphedema, there's no medical therapies currently available. So we're looking to bring treatments for these diseases into the 21st century. Next slide, Adam. Our pipeline has two franchises, a neurological franchise and a lymphatic franchise. In the neurological franchise, CDNF is our lead asset that recently completed a phase 1, and now we're moving on to further development with alternative and expanded routes of administration. We also have X-CDNF that is currently in research, and that is due for compound selection early next year, and I'll touch on all of these later in the presentation. And then for our lymphatic franchise, lymphactin for breast cancer-related lymphedema is our lead asset that is currently completing a phase 2 study. We also have research in early research in other lymphatic diseases. And the common theme between these two franchises is the regenerative medicine, the disease-modifying science. And success with CDNF or X-CDNF will give us access to a multi-billion euro, multi-billion dollar market. And similarly for lymphactin, in our estimates, will give us access to a market worth at least 600 million euros. Next slide, Adam. Okay, next slide. So spending some time on Parkinson's and CDNF and X-CDNF. Now, the total neurodegenerative disease market is massive. It's going to be 63 billion, estimated in 2026, currently 35 billion. Parkinson's, currently there's 10 million patients worldwide, and the market size for Parkinson's is approaching 6 billion. Our approach is the current therapies are symptomatic and don't really manage the disease per se. Our approach with CDNF is a natural molecule, occurs naturally in the body, and its natural role is to protect neurons in the body. So we're looking to harness that natural mechanism of CDNF and modify Parkinson's and other neurodegenerative diseases. Next slide. It's worthwhile spending some time on the mechanism of action, and I'll go through this fairly in a structured way. It looks complicated, but it's actually fairly straightforward. So the key word here is proteostasis, and proteins are the building blocks of the body. If you mess with proteins, you mess with the body. Simple as that. If you're on a diet or looking for a diet, they'll always say cut out fat or cut out carbohydrates, but they never say cut out proteins, because proteins are so fundamental to the body. And the process or the pathway that ensures proteins are maintained in the body is called proteostasis. And there's three key components to this. The one is the synthesis, or the production of proteins in the cells. The second one is the folding, which is proteins need to be folded in a certain three-dimensional structure to exert their effect. And the third one is degradation. So it's like a quality control, because if proteins are faulty, or if there's too many proteins, then the degradation can take care of those before they do damage in the cell. Now, in diseases like Parkinson's and other neurodegenerative diseases, proteostasis goes wrong. It goes faulty and it becomes awry. And that's where CDNF steps in. First of all, it acts to ensure that the synthesis and the production and the making of the proteins is maintained and correct. Second of all, it ensures that the folding of those proteins is absolutely spot on. Without that folding, there's going to be damage and the cell will die. And the third aspect is it steps in to ensure that the quality of the proteins are correct and maintained. Now, when proteostasis goes wrong, it also causes collateral damage via inflammation. And CDNF also steps in to that side of the equation by controlling the inflammation around the cell and in the tissues to prevent further damage. So 4 key mechanisms that CDNF acts on this very important proteostasis pathway. Next slide, Adam. So, and proteostasis occurs in an organelle called the endoplasmic reticulum. And this is the orange, light brown, folded organelle that you see in the picture there. And you can appreciate that it's a very big organelle. It's got a huge surface area with all these folds, and it covers the nucleus entirely. So it probably occupies more than half the cell. You multiply this by billions and billions of cells in the body, and you start to understand what an important role proteostasis and the endoplasmic reticulum has. And that's exactly where CDNF acts, on proteostasis in the endoplasmic reticulum. So remember the word proteostasis. I'll come back to it later. Next slide, Adam. So based on the mechanism of action and some compelling preclinical data, we took CDNF into the clinic, into a study that completed recently. It started off with 6 months. Patients were put onto placebo or CDNF. There then was a further 6 months where the placebo patients were also put onto drug, and currently the study is in its follow-up phases. The characteristics of the patients that entered the study were on average about 60 years old, and importantly, they had very advanced disease, at least 10 years since disease onset, the first motor symptoms. And that's important because these are very, very advanced patients where their dopaminergic neurons are no longer present. They are lost. Next slide. We were pleased in early this year in Q3 to announce that the study had met its primary endpoint of safety and tolerability. The adverse effects were mild and transient. There was a similar safety profile in the first 6 months compared to the second 6 months. There was similar safety profile between the groups. And importantly, there were no dose-limiting toxicities related to CDNF. Now, this was not an efficacy study because the patients were so advanced, as I mentioned, other than to say that we saw no worsening of disease in these patients, in a disease that you would expect to worsen over the course of the study. Next slide, Adam. So with that as a solid foundation, the phase 1, we're now building for the future for a successful CDNF program. In the first phase 1, CDNF was administered via a surgical drug device combination that was inserted into the brain, very complicated, very invasive. We're now moving away from that to back CDNF as a standalone product to a more patient-friendly, regulatory-friendly, physician-friendly route of administration, specifically nose to brain, so intranasal in the nose via a device, and skin injections, so just a subcutaneous skin injection. Now, there's solid scientific merit and rationale for both of these approaches. We have data with CDNF using these administration options, showing that we can cross the blood-brain barrier, number one, and we can do so in therapeutic levels, number two. So this is an ongoing, these programs have been ongoing, and we're backing CDNF as a standalone product without having to rely on surgery. So that's the first component of the program moving forward. The second component is we want to accelerate development. So the fact that we no longer have the drug device, the complicated or invasive device in the equation, means that we can accelerate and increase the chances of successful development, approval, and commercialization. And as part of that, it's our hope as well that we will accelerate potential partnering transactions with CDNF. And the third key pillar of the program moving forward is going to be targeting earlier stage patients. So as I mentioned, the patients in the phase 1 were advanced with no dopamine neurons. The dopamine neurons were lost. CDNF functions on remaining dopaminergic neurons that are viable. So while the dopamine neurons are reducing or dying, CDNF can come in there, turn things around, and do the job. But once the dopamine neurons are dead or lost, then it's too late. So we're going to be expanding the target population to an earlier stage in the disease. Next slide, Adam. What about ex-CDNF? So that's CDNF. Let me tell you a bit about ex-CDNF. So this is a second asset in Parkinson's disease. And what we've done here is we've taken the smallest, most potent fragments of the parent CDNF molecule, and we've engineered that to achieve two things. One is to be able to cross the blood-brain barrier, and two, to retain the potency. So still be able to exert the efficacious effect of CDNF after crossing the blood-brain barrier. And we've engineered the molecule to achieve specifically those two criteria. Next slide, Adam. So here's some data on the potency, and I'll walk you through it. So the plot on the left-hand side on the y-axis is so that's tyrosine hydroxylase cells. So this is basically dopaminergic neurons. These are the neurons that go wrong in Parkinson's. And then along the x-axis is concentration. And what we see, the higher the number of the cells, the better, because we see in control animals here, this was done in rats, in control animals here, who didn't receive therapy, the number of surviving cells is low, of the surviving tyrosine hydroxylase, the surviving dopamine neurons are low. Once we treat them with CDNF and ex-CDNF, we see the number of surviving cells increased by all orders of magnitude. And this is only with 48 hours of treatment, so a very short, brief treatment. And we see here, in fact, ex-CDNF could be 10x fold more potent than CDNF. So tick in that box, we need to retain the potency with the fragments. On the right-hand side here, what we're looking at is alpha-synuclein, and that's one of the bad proteins that I mentioned in the degradation, the quality of control, that we need to get rid of. So here it's the opposite. Here in these cells, those levels of alpha-synuclein are very high in the red at the top. Once we treat them with CDNF and ex-CDNF, we see that those levels in the cells drop precipitously. Again, 48 hours of treatment, and we see these results. So again, the efficacy is established compared to controls, and another tick in the box for potency. Next slide, Adam. What about crossing the blood-brain barrier? So here we see on the left-hand side on the y-axis is just the amount in plasma, and on the x-axis is time in minutes. And that's the same on the right-hand side. On the left-hand side, we just see the profile in plasma. So as the rat is injected, it goes up and down as you would expect. And then what we see on the right-hand side, this is actually looking at interstitial fluid inside the brain. So this is on the other side of the blood-brain barrier. So ex-CDNF has crossed the blood-brain barrier, And once it's there, it's stuck around for a very long time. And all the time that it's there, it can do its job and act on the dopaminergic neurons. So very important to see here, number one, that we cross the blood-brain barrier, and number two, we maintain the concentration once we're there. Next slide, Adam. So just to summarize that, there's three key aspects to the program moving forward, where we're combining the ability to cross the blood-brain barrier as well as the potency, and that is summarized in CDNF, the intranasal program, CDNF, the systemic or skin injection program, and X-CDNF, which I've just gone through. Next slide, Adam. I said I'd come back to the word proteostasis, and this is why, because we are in great company in this space. It's a very hot topic at the moment. It's an important research area in diseases, specifically neurodegenerative diseases. We have Prothena and Roche, market cap of half a billion. They entered into a deal for $600 million. We have Humanity, a similar deal with Merck announced earlier this year. We have Calico Labs, which is a Google-backed company that injected $2.5 billion into research in this area. And then we have Denali, who have a market cap of $5 billion. And all of these companies, importantly, all of these companies act on a specific part of proteostasis, whereas for Herantis, we act on the entire pathway, as I've taken you through. So we're in great company with these other companies, the collaborators, and it's certainly an important focus for the biotech industry currently. Next slide, Adam. I think in the interest of time, let's go to lymphactin. Okay, I'll talk about our second disease area, that's lymphedema with lymphactin. Next slide. So lymphedema is the lymphatics, all the lymphatic vessels in the body. Their function is to drain fluid and water back from the extremities, the legs and arms, back into the heart, so that fluid and water can be reused. Now, in lymphedema, something goes wrong with the lymphatics. It's either genetic, or there's due to infection, or due to injury, or some other insult, with the result that the lymphatics are dysfunctional and the fluid doesn't drain. And the final outcome is that those limbs where the lymphatics are supposed to drain just become laden with water, heavy with water and fluid, very uncomfortable, very painful for these patients. And another medical term for lymphedema is elephantiasis. And that's because the limbs that are affected by this literally look like an elephant's limbs or arms, because they are so big and heavy and soggy with water, because there's nowhere for the water or fluid to go. Our focus is on breast cancer related lymphedema. So patients come in for breast cancer surgery, and at the same time, they often have the lymph nodes removed in their axilla, in their armpit, which means that these lymphatics in the arm are no longer joined to the lymphatics of the body. So the fluid collects in the arm. The arm becomes very heavy, sometimes 5 or 10 kilograms just for an arm, which is very painful, very debilitating for these patients. And there's no therapy for this other than putting a giant blood pressure decompressive cuff. 30% of breast cancer patients who have a mastectomy develop breast cancer-related lymphedema, and our focus with the lymphactin is to reestablish the lymphatic drainage system so that the lymph can drain from the extremities back into the body and relieve the suffering for the patients. Next slide. A quick word on the mechanism of action. So VEGF-C, or vascular endothelial growth factor, works on two main compartments or components of the body. One is the blood vessels. It's a very potent growth promoter of blood vessels. And the other one has been a similarly potent growth promoter of lymphatic vessels. And for us, with VEGF-C and lymphactin, we are specifically focusing on lymphatics. We know the receptor, we know the interaction, and that is our focus with lymphactin, purely on the lymphatics. Next slide. This just summarizes, so you can see the top graph, there's no connection between the lymphatics in the arm and lymphatics in the body. And in the bottom graphic, that's what lymphactin looks to reestablish those connections between the lymphatics in the arm, lymphatics in the body, such that the lymph and the water can drain out of the arm and relieve the suffering of these patients. Next slide. Where are we? Well, we've completed a phase 1 successfully, and we just announced 24-month data on the phase 1. It continues to be safe and well tolerated. And although this was not an efficacy study because there was no control group, we did see volume reduction in approximately 50% of patients. We did see improved lymphatic flow in some patients, as well as improved quality of life in most patients, which is a very, very important indicator for patients as well as for regulators. We're currently in a phase 2 study, and we expect that to read out early next year, and that will give a clear picture of efficacy because there is a control group that got Lymfactin and others that didn't get Lymfactin. Sorry.
Craig, sorry to cut in. I think in the interest of time,
Sure.
we'll move over to a little bit of a Q&A just to finish off. So,
just the summary slide, that's the next slide. You just leave it there.
yeah, okay, we can leave that. We can leave that up. There we go. So my first question is around the CDNF program and the switch to alternative routes of administration. And I was wondering if you could add any detail on kind of the prospects of achieving sufficient blood-brain barrier penetration with these alternative routes of administration in the light of the fact that obviously intracerebral delivery was pursued initially. Can you give any more details around that?
Yeah, sure. So there's quite an echo. I don't know if that's me or... Can you hear me okay?
We're adjusting the sounds here. Is that better now?
Yes, that's good. That's good. So as I mentioned, the three follow-on tiers of CDNF. Number 1 is intranasal, and with intranasal we already have data that we can cross the blood-brain barrier, number 1, and number 2, we can cross it at therapeutic level. So we already have that data. This has been an ongoing program for a couple of years, and right now the focus is on the formulation side of it to even increase that even more, with a target roughly 10% to cross the blood-brain barrier and get into the brain. So that is a existing program that we know we can cross the blood-brain barrier. The second one is the subcutaneous route. Again, it's a preclinical program focused on acute neurodegenerative disease where the blood-brain barrier is actually a bit compromised. And there, again, we know that we can cross the blood-brain barrier in meaningful amounts in order to be able to do the job. And the third key program is the xCDNF, the small one, where we know that we've engineered those particles small enough to cross the blood-brain barrier in therapeutic levels. So we've established all of that we can do it. Now it's a question of optimizing it and making sure that we can get it to levels that can do the job in the brain. Does that answer your question?
Yeah, no, that's great. Thank you very much. On that same note, with the shift to alternative means of administration, intranasally and subcutaneously, can you give any details around when we might expect to hear more about the development path forward for CDNF specifically, timelines and such? Sure.
So clearly we're working through the different programs, but in summary, or to give a broad framework, next year will be spent on the preclinical development of the new formulation routes, as well as for xCDNF. Thereafter, the following year will be spent on IND enabling in order to get into the clinic. So once we've done the IND enabling, we'd expect to get back into the clinic thereafter. Okay, great. And we've, as you mentioned in the presentation, obviously seen the long-term follow-up data from the Lymfactin phase 1 study. We've got the phase 2 readout early next year in the first quarter. I was wondering if you can give any more details on what the development path will look like post those results, assuming obviously that they read out positively. What can we expect in terms of subsequent studies and the design of those studies and so? Yes, I think with the Lymfactin, it's quite well mapped out. Pending data, as you say, assuming that the data is positive, then the next logical step would be to do a phase 3, a pivotal phase 3 study. So that would be something that we could expect in 2023. Mm-hmm. But as I say, pending data. So we haven't sort of communicated anything. That's clearly a distinct possibility, but we need to wait and see what the data shows. Great. And just lastly, I think in the interest of time, what can you say about the cash position of Herantis, specifically around the potential need for
cash infusions to continue the development programs? Are there any details that you can give around that?
Yeah, so I think what we've disclosed is that we have runway through to the beginning of the second half next year, end of the first half next year.
Okay, great. I think that's all the time we have. So thank you very much, Craig, for the presentation, and thank you to all our listeners.
Thanks, everybody. Thank you.